Hi class, this is the Module 14 lecture. The topic is molecular biology. All right, so to begin, we're just looking here at a picture of the measles virus. I could have put any number of viruses here. And of course, now we're probably thinking about the virus that's responsible for the pandemic, the SARS-2 COVID-19 virus. So this is not that. You've all seen plenty of pictures of that virus. The reason why I'm introducing the topic of molecular biology with a virus is actually for a few reasons. First of all, viruses are actually used in molecular biology as a tool. Much of the work I did in my research lab at University of Arizona could not have been done without certain viruses. We also need the tools of molecular biology really to understand them and to even visualize them. So this is a very powerful type of microscope. Electron microscopes are what are used to visualize viruses. And fortunately, there's not really much measles. It doesn't affect our world the way it used to, and that's due to the vaccination campaigns for measles that have been around for decades now. So measles is considered to be a highly contagious virus, and it's spread through the respiratory route, similar to the SARS-2 virus. So a lot of people don't understand that. They think that it's spread through the skin because the skin can be involved in, producing a characteristic type of rash. And it's true, it can be spread through contaminated pus and material that's in those blisters, but its primary route of transmission is the respiratory route. We've had a number of outbreaks in California. There was one in 2014 tied to, I think it was to Disneyland. And that actually came as a surprise because it was one of the big outbreaks again. It was considered to have been eradicated really from our country due to the successful vaccine campaigns. So as I began the topic with, to study any virus, and certainly to be able to produce vaccines and then test those vaccines, that all sits right in the field of molecular biology. And it would occur at the different levels. So at the level of the protein, as well as at the level of the nucleic acid. All right, so we're going to go through the first 3 sections. This is from chapter 10 of your textbook. I had to think about that for a moment. All right, so let's begin with the structure of genetic material. And some of this is familiar to us. This is probably new. Here we're looking at a different type of virus. This is called a bacteriophage. This one is called phage T4, that's its name. So on the left side, in this pretty green and yellow picture, this is an electron micrograph. So it's an actual image of the virus. And then we've got the cartoon depiction of it on the right. And these are viruses that infect bacteria. So that's why they are called bacteriophage. Phage means to eat. So they eat the bacteria, they consume it and kill it. Really everything about the structure of this virus is tied to its function. We've seen that theme now over and over in biology. I think it looks like a spaceship, and the way it operates is kind of like that. So this virus does not enter the bacteria. It does not go completely inside. What happens is it docks down on the surface of the bacteria. And this is a specific relationship. Certain bacteriophage are associated with infecting a specific type of bacteria. So it finds the right bacteria and lands down on it, kind of like a spaceship would dock. And it uses the tail fibers for that purpose to help anchor it onto the surface of the bacteria. And after it's anchored down through its tail fibers, the tail fibers spread out. And then what happens is the rest of the virus lands, kind of lowers down. So it's in contact with the surface of the bacteria. And you can see there's little spikes here that are for that purpose. And then what happens, the DNA that's inside the head region of this virus, it is what goes inside. And it's similar to how a needle would work, where the DNA goes through the surface of the bacteria, or a syringe would go through the surface of our skin. So only the DNA goes inside, and the rest of it stays on the outside. And what will happen is these bacteria will become infected with a quantity of these bacteriophage that are replicating and replicating and replicating. And eventually the bacteria bust open, and then out come a bunch of new phage that can go on to infect other bacteria. So here's that replication cycle. So we've got the bacteriophage. And again, these are somewhat matched relationships. I don't want to say that they're really specific, but just like we have certain viruses that infect us, and our dogs and cats are infected by other viruses, they don't usually go back and forth between different species. It's kind of like that here. These are just a class of virus that infect certain bacteria. So it's docking down, it's attaching with its tail fibers to the surface of the bacteria. And now you can see the phage DNA is being injected through the surface of the bacterial cell wall. And it takes over control of the bacteria then. Everything happening inside for the replication and life cycle of the bacteria is now about making new phage. So the phage DNA has to be replicated, but also the process of transcription and translation needs to happen so that the phage has all of the different proteins so that it can assemble as a full phage molecule. And then it will be able to go on and infect other bacteria. So this is the process of lysis, where the cell is being released and then out come all of those new phage. So when we're thinking about replication here, we have to think about replication of the nucleic material, the DNA, as well as protein synthesis to make the tail fibers and the docking material, as well as the head of the phage that houses the DNA. All right. Well, you've probably heard of Watson and Crick as people that discovered DNA, and they were awarded the Nobel Prize along with another person. But Rosalind Franklin really is responsible for having discovered the structure of DNA. She was a radiologist. She was the woman who actually got the very first picture of the DNA molecule. Unfortunately, she died prematurely. She died young, and that's because she died of radiation exposure doing her work. And she did not win the Nobel Prize because Nobel Prizes aren't awarded posthumously. So in some circles, she's unrecognized for her very vital work and selfless work in discovering the structure of DNA as a double-stranded helix. So here she is, and then here's a picture of the image that she captured that showed that it's double-stranded, and you can probably appreciate that it's a double-stranded molecule. So that was taken by X-ray. Okay, so looking at the structure of the DNA molecule. So there's a lot here, and it looks busy, but let's first of all begin with what we already know. Let's focus on the monomer of the DNA polynucleotide. So we've got, so this goes all the way back to earlier in the semester. A nucleotide has three components. It has a sugar, it has a phosphate group, and then it has a nitrogenous base. So this makes up this, the structure composed of these three things makes up the nucleotide of DNA here. And then these are strung together to create our polynucleotide, our chain of DNA. But then DNA is double-stranded, and it exists as a what is called a double-stranded alpha helix. And there is chemical bonding that's happening between the two chains of DNA that hold it together. So we're going to have to dig into all of this to understand the topic of molecular biology. Now, the sugar for DNA is deoxyribose. That's where the D comes from in DNA. Whereas in RNA, it is ribose. And then we've got four nitrogenous bases or nucleobases. In DNA, it is C, G, A, and then T, thymine. Now, RNA has the first three, C, G, and A, but there is no thymine in the RNA molecule. Instead, it is replaced with uracil, which is abbreviated with the capital U. So that's one of the ways you can look at a sequence pretty quickly and see if it's DNA or RNA, is you look for either the presence of T's or U's. Now, if the sequence doesn't have either, then you don't know. And then we've got the difference in the sugar, deoxyribose versus ribose. And then we've got a third feature that's different between DNA and RNA, and that's not really shown completely on this slide. But what that is that DNA is double-stranded. RNA doesn't exist that way in our body. There's no such thing as double-stranded RNA at any time, even during replication of anything. So we never have double-stranded RNA in our body. In fact, if we do, it means there's a virus. There are certain viruses that have double-stranded RNA genomes, and our immune system actually knows that and will become activated to fight a virus infection if it recognizes double-stranded RNA. So those are the three main differences between DNA and RNA. All right, so the thymine and cytosine, these are the pyrimidines. Notice they only have one ring that composes the molecule, where adenine and guanine, are each made out of two rings. They are the purines. The way that base pairing happens is thymine goes with adenine. That's why they're both shown as green in this figure. And then cytosine base pairs with guanine. They're both shown as orange for that reason. So that means that the DNA double helix, it's roughly the width of three of these rings across, because thymine has one, and then it base pairs with adenine 2, 3. The same is true for cytosine, 1, pairing with guanine 2, 3. So that's it's actually a signal to our body knows that the DNA is being made correctly during DNA replication if the width stays consistent, because it shouldn't. If thymine bonds with guanine, that's not a correct base pair match, and the enzymes that are used for DNA replication, well, they're really efficient. They actually figure this out and usually correct it for us. And that's why most of us don't have cancer. So looking at that a little bit more, adenine base pairs with thymine, and then guanine base pairs with cytosine. A to T, C to G in DNA. There are 2 hydrogen bonds between adenine and thymine. For the other base pairs, for guanine and cytosine, there are 3 hydrogen bonds that form between the 2 base pairs. So there's a slight difference. So let's also take a moment to think about what a hydrogen bond means in this context. Going back to the chemistry we learned earlier in the semester, hydrogen bonds are weak. They're the weakest type of chemical bond that in biology. And that's necessary in order for DNA to replicate. It actually has to come apart in order to do DNA replication. The DNA helix has to separate. If the two chains were held together with those strong covalent bonds, that would be very challenging to separate the molecule. Now, there are covalent bonds in the DNA molecule, but they're just not between the nitrogenous bases. They are on the sugar phosphate backbone. So the shown here in the yellow and blue, we've got our phosphate group and then the sugar. That is where the covalent bonds that hold a single DNA strand or RNA strand, they're located in the sugar phosphate backbone to maintain the integrity of the sequence. The hydrogen bonds are what hold the two chains together. But these type of bonds fit the right function of the different parts of the DNA molecule. We want our strand to be held together with strong covalent bonds, or else we'd have mutations. So looking at it again, the sugar phosphate backbone, and there's two of them because it's double-stranded, that is where the covalent bonds are, in between the sugar, the blue part, to the phosphate group, and then again, from the phosphate group to the next sugar. So looking at our partial chemical structure in the center, we can see the sugar phosphate backbone. Those are covalent bonds on both sides. So think of it like a ladder. I like to think of the double-stranded DNA molecule as a ladder, where the sides of the ladder are the individual chains of DNA, and they're held together with the strong covalent bonds. And then the rungs of the ladder, these are the hydrogen bonds in between the matched nucleobases. And these are temporary bonds, just like we learned about at the beginning of the semester when we were studying the chemical properties of water. Hydrogen bonds are temporary. They're constantly making and breaking. Now, I actually like the computer model quite a lot because I did a lot of work on the protein level. It shows you the space that is occupied by each of the atoms that make up these molecules. So you can again still see the sugar phosphate backbone, but I think you're able to better appreciate the size of the molecule. The other two are helpful so that we can look at the chemistry. So those are the three ways that we can look at DNA. All right, let me get this video playing for you. Oh, it jumped off.
DNA and RNA structure. DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. This is a close-up of one DNA nucleotide. A DNA nucleotide is composed of a nitrogenous base, phosphate group, and a deoxyribose sugar. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide's sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' directions. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains or strands are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, well, that video went into some topics that we are going to talk about in the remainder of this module. So please do take a look at this video at other times, because the material in it is important and it's explained. It's always nice to have a different explanation to things, but this is part of the material that you'll need to understand for this chapter or for this module. So that way you've got it to go back and look at. It's a pretty comprehensive video. Okay, so again, we can think of the sugar phosphate backbone like the sides of the ladder. These are held with covalent bonds. And then the hydrogen bonds form between the matched base pairs. Those are like the rungs of the ladder. But then the molecule twists. Wow, that was something just shook my windows. Sorry about that. Then the molecule twists once and then again. So it's actually it creates a supercoil in the molecule, and it is the double-stranded alpha helix that is the correct structure of DNA. All right, so now we're going to go through the process of DNA replication. We'll talk about some of the terms that were mentioned in that video, specifically the 3' and 5' end of DNA. Those are important, especially as we begin to talk the language of DNA replication. So when we begin, we are talking about let's put some of our genetic terms to use here, like that we learned in the last module. So our DNA molecule is the parent DNA molecule. It is used as a template, as a source of instructions, if you will, to make new strands of DNA during the process of DNA replication. So the parent strand is shown in blue, and those hydrogen bonds that hold together the base pairs, they come apart. We'll talk about how that happens, but they come apart in DNA replication. So the double helix, the double-stranded molecule separates, not the whole thing at once, just pieces of it at a time. But now we have the new daughter DNA strands being assembled. They follow the base pair matching. So the reason why the T goes here is because T always goes with A. G goes with C. So it begins by having really like a pot full of free nucleotides, and they match and fill in following the template of the parent. And then they are stitched together to make the continuous chain. So at the end of DNA replication, we are left with two molecules of DNA, and they're the same. Now, each molecule has one parent strand in blue and a newly synthesized DNA strand, daughter DNA, shown here in gray. So we consider DNA to be semi-conservative. What that means is that the new DNA, it's semi-conservative. So that means it's got part of the old and part of the new. So now we're looking at, digging a little bit deeper, looking at how this process happens. So up top in this figure, the parent molecule is still assembled in the double-stranded alpha helix, but it does come apart during DNA replication. And now we can see how the daughter molecules are filling in to create the two newly synthesized strands of DNA. So this happens as the parent strand untwists, and there's actually an enzyme that does that. All right, so let's take a look at this overview of DNA replication. I'll play it start to finish, and then we'll come back and dissect it together and go through the different components.
DNA replication, an overview. In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules. Here's a simplified close-up of replication, showing what happens along one of the template DNA strands. Using a supply of free nucleotides, DNA polymerase assembles a new DNA strand along the template strand, following the base pairing rules. Adenine pairs with thymine and cytosine pairs with guanine. Now let's look at the DNA replication process. Replication of a DNA molecule begins at a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we've untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical daughter DNA molecules. Now, let's review the synthesis of DNA by assembling a new DNA strand. We'll use the top DNA strand as the template and choose from the free nucleotides below. Note that D stands for deoxyribose and R stands for ribose. We're only using the deoxyribose nucleotides because we're building DNA, deoxyribonucleic acid.
Okay, so there's our process of DNA replication, really start to finish. So now let's go through that ourselves so that we can understand the process, but also what these different terms, what they mean. So just to clarify here, 3 phosphates. You may know more about this molecule than you think just looking at it. We know about energy. We know about metabolism now. 3 phosphates means that this has an energy source, since those phosphates can be used in the production of ATP, and then that ATP can be harnessed for energy. So I just wanted to tie in some more about the structure and function of the molecule here. Okay, so what the video ran us through is when we untwist the DNA helix, so we've got it just stretched out, untwisted, there are origins of replication. So these are certain sequences in the DNA sequence, and there's multiple of them. But these are the parts where the enzymes that are associated with DNA replication, it gives them a place to bind so that they can begin their job. So what happens is the origin of replications, these are where the replication bubbles are. So the strand has separated. Those hydrogen bonds holding together the 2 strands of parental DNA have come apart, and we are synthesizing the daughter DNA, moving in both directions away from the origins of replication. Eventually, the sites grow together, and we are then left with the 2 strands of DNA, each containing a parent strand and each containing a newly synthesized daughter strand of DNA. Semi-conservative. Okay, so now let's talk about what this 5' and 3' and polarity of DNA is. And we do need to understand what this means to really be able to picture and understand how DNA replication happens. So we've got our sugar phosphate backbone of both sides of our DNA double-stranded molecule. We're looking at just a sequence of 4 nucleotides, so just a tiny little piece of DNA here. And then you can see the hydrogen or the base pairs are bonded to assemble our molecule. What the 5' end of DNA is it's where the free phosphate group is. And I don't mean that the phosphate group is just floating out on its own. What it means is that it does not have a covalent bond to another nucleotide. The strand ends here. So the end that has the phosphate group is 5'. The end that has the hydroxyl group, OH, is always 3'. So the 2 strands are opposite in polarity. It goes 5 to 3 on one strand, and then the next strand is 5 to 3, but in an opposite orientation. So they're complementary because of the base pairing, A to T, C to G. And the sequences are indeed the mirror image of each other all the way down the molecule. Anti-parallel refers to the prime of the ending, where the 5' end is associated with the 3' end of its other strand of DNA. Now, the reason why this is important is because the main enzyme that's involved in making the new DNA polymerase, it can only move in a certain direction. So that means we do have a slightly different process depending upon which strand of DNA we're focusing on during replication. So it's slightly different. Okay, so let's look a little bit more. This actually comes out of a different textbook. So it's a textbook that I use in microbiology. So our 3' end has the hydroxyl group free, and then the 5' end has a phosphate group free. So we're looking at our DNA double-stranded molecule, 1, 2, 3, 4 nucleotides that are still held together by those base pairs, by those hydrogen bonds. But now let's look at the step of making more. So our template, so this is the parent strand here, our template has an A in this position. So that does mean that thymine is what comes in next in the growing new strand of DNA. So the new strand of DNA it always begins at the 5' end, and then we are adding on at the 3' end. Okay, that's a lot. Let me try to back that up a moment. So here comes our incoming nucleotide, which is a thymine. And now look over at the product. DNA polymerase has added in this thymine onto the 3' end of the newly synthesized daughter strand of DNA, because that is the way DNA polymerase works. Let's look at the name. It's a polymerase. So ACE tells it's an enzyme that is making a polymer of DNA. That's what DNA polymerase does. And we do have a few different types of them. And if you go on to a molecular biology class, you'll learn about the different types then. For our purposes, we understand that it is the enzyme that adds on the nucleotides to make the new strand of daughter DNA. So it's only happening at that 3' end. And the reason why is because there needs to be the opportunity for the covalent bond to form in between the existing end of the DNA molecule and what's coming on next. So that's happening through these phosphates and sugar molecules, again, on the kind of the stand side of the ladder, if you will. So 2 phosphates leave, and then we have a covalent bond formed at the 3' end of the newly synthesized strand. Okay, so our parent strands, we've got on the top sequence is 5 to 3. So now let's see what happens. Let's look at the differences between DNA replication in these 2 strands. DNA polymerase only adds on the 3' end of DNA. So that is this strand up top in this case, because it's anti-parallel. It begins with 5. Okay, good. So DNA polymerase can move right along because it's always growing on the 3' end. It can't grow on the 5' end. It just stops there. The chemistry is not right on the other side of the molecule to facilitate the incoming nucleotide. So the nucleotide's always going to come in and bond on that 3' end. So the newly synthesized strands grow by adding on 3' end. Now, so if we are the 5 to 3 parent, that's great, because that means the daughter strand is going to begin with 5'. But now let's look at the example of the 3' to 5' template of the parent template DNA. Sorry about that. So it can only do the same thing. It has to grow starting at the 5' end and then adds on 3. But our template strand does not have the chemistry set up this way. Notice that the template strand now is beginning with 3. But DNA polymerase doesn't work that way. It has to begin with 5, and then we add on at the 3 end. So what ends up happening is we have one strand that's made continuously as one piece, and then the other strand is actually made in a bunch of little pieces. And then those little pieces have to be stitched together with another enzyme. So one strand is quicker than the other. DNA ligase, let's break down that name. ASE tells us it's an enzyme that ligates, that means to sew or to stitch DNA. So it takes these pieces and stitches them together to make one continuous piece of DNA. So the overall direction of replication of the daughter strands in this example is starting here and going this way. We're following that leading strand, the strand that goes continuously. But even on this lagging strand, it's still starting 5 and then adding on at 3. It just does it in a bunch of pieces, and then other enzymes come in and help to put it together. So this can be tricky to understand, but the key piece of information is that DNA polymerase adds on to the 3 end. So this is just from that other figure, just to give you another way of looking at it. We have our double-stranded DNA molecule. So now we've got some of the other enzymes shown here. So the top strand is 5 to 3. The bottom strand here is 3 to 5. So that means that we're going to have, again, a difference. So it's the bottom strand that is going to be continuous now, because the template is 5 to 3 on the parent. Now you have to go opposite. So that means the daughter is starting with 5 and is growing on the 3, which is right. So we get one continuous piece. So we have what's called an RNA primer, this yellow bit, that is synthesized by an enzyme, and it's a point of attachment for DNA polymerase, so that it knows where to begin and can do its job, adding on at the 3' end. So on the lagging strand, this is happening over and over. There are multiple RNA primers because it is synthesized in many different little pieces. And that keeps it, technically, it's still, look where the primer is. The primer is on the 5' end. So the new DNA here is 3 here. So that means it's 5 here. And the primer should be at the 5 end, because DNA polymerase adds to the 5 at the 3 end. I know that's confusing. Okay, so what's happening is it moves down the DNA strand, but it's also happening at multiple places at the same time. Our genome's just too big. If it did everything one at a time, we would never be done replicating. All right, I'm gonna play a couple of videos here because I know that these are challenging topics, and I'm not trying to not explain it to you myself. I'm just want to give you alternate materials so that you can study. Okay, here we go. So now we're just looking at the leading strand of DNA synthesis. So that means the parent, I'll leave that to you. Is the parent strand going to be 5 to 3 or 3 to 5? Oh, where's the start? Here we go.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adenine always pairs with thymine. Guanine always pairs with cytosine. Adding new nucleotides works the same way on both the leading and lagging strands.
Okay, let's move on to the lagging strand.
Each piece of the lagging strand begins with a short segment of RNA, shown in red. A protein clamps the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together.
So the lagging strand does take more time because it has more work to do. And it also uses more materials. Okay, so now we've got, just focusing on the replication, we've got our parent strand down at the bottom. So the top sequence in the green is 5 to 3. The bottom sequence is 3 to 5. It's not written on there, but you know that this bottom strand here, this is the 5 end this is the 3 end because this end is 5 because it's anti-parallel. Okay, so the top sequence is 5 to 3. This one is 3 here and 5 here. So we're just looking at the lagging strand here. We're leaving out the leading strand process because it's been done in one fell swoop. So here is the pieces of RNA. These are primers. They're called primers. Here's the word, RNA primer. And notice there's multiple pieces along the lagging strand. These are just small pieces of RNA, but it is where this other enzyme called primase, make a primer, that's what it's doing, make prime, if you look at the word, it is able to attach there to insert the, to create that primer. So it's what makes it. So here's our DNA polymerase, and it's moving along 5 to 3. It's moving this way because here's the template of the parent, 5 to 3. That means it's 5 prime starting here as we make our lagging strand. So here's 5, here's 5, here's 5, and then finally we get to the 3 prime end. And then the other enzyme that we were speaking of is DNA ligase. Its job is simply to stitch together the newly formed pieces of DNA. They're not joined together because those enzymes keep having to jump off because they can only go in one direction. So it's kind of like that old saying that you take 2 steps forward to go 1 step back. That's what the lagging strand reminds me of. It's happening, but we keep having to go in reverse so that we are in the right direction to go a little bit forward. Because again, that enzyme can only add on the 3 prime end. It cannot add into the 5 prime end. Now these little pieces, here's the name of what they're called, Okazaki fragments. And that is after the person who discovered them, who first identified the process of lagging strand synthesis. All right. Hopefully, I know you'll have to spend some time with those concepts to put them together. All right, so now we're going to move to our next section, and we're going to look at the protein synthesis side of it. We've been talking about just DNA replication, but if we're talking about a cell or a virus that has to replicate, it's not just nucleic acid, right? We're made of our DNA, the RNA, and protein. So we've got our 2 processes. Transcription is DNA to RNA, And then translation is RNA to protein. So the central dogma of molecular biology is the flow of information goes from DNA through an intermediate of RNA to protein. Every organism on Earth does it this way. There are some viruses that don't, but they're not organisms anyway. Viruses are intracellular parasites. They are not organisms. They don't meet the definitions of life because they can't replicate on their own. But we do have a couple categories of viruses that can go in opposite direction. And probably most famously, that would be the retroviruses, of which human immunodeficiency virus belongs. All right, so here's our central dogma of molecular biology. It begins the flow of information. So this is another core theme of biology, the flow of information. It begins from the DNA, which is organized in the form of chromosomes when our cells are not undergoing replication. So transcription begins with the DNA template. It ends in the process, in the production, I should say, of RNA. Notice this RNA molecule is single-stranded. We do not make double-stranded RNA in our body, never. It's a flag that something's wrong if it's there. So transcription has to happen in the nucleus because that is where the DNA is. And how can we copy the DNA if we're not where the DNA is? So location is really important in life too, in real life, not just property. Next process is we begin with this RNA molecule that actually does come out of the nucleus. I'll show you that in a moment. And it is made into protein through the process of translation. So that is the flow of information. It's also called, when we're just talking about the protein side of it, we call that protein synthesis. And there are two steps to it, transcription and then translation. So looking at our DNA molecule, in this example, you can see that there are different colors and those represent different locations on the DNA, so different genes. So we've got 3 genes here, and we're looking at a portion of gene 3. And we're looking at the DNA. So we're just looking at one stranded DNA here, right? Notice the little black box is not on the double-stranded molecule. It's just on one strand. So we're only looking at one template of DNA here. And it uses the sequence of DNA to make a messenger. So this is the RNA in pink. And it follows the same base pairing rules that we learned about for DNA, with the exception that there is no such thing as thymine. There's no T in RNA. So A would go with T if it were DNA, but it's not. This is RNA, so it goes with U. A goes to U, A goes to U. C and G are still the same, but then A goes to U. So transcription is creation of this RNA molecule using the DNA as a template through the anti-parallel complementary base pairing rules. The RNA is organized in what are called codons, and these are units of 3 RNA nucleotides. And those are what are read through translation to be made into the respective amino acids. We have 20 amino acids, but every time we see a codon, it should make the same thing every time. So notice here we have UUU and it makes a square. Well, down here we have UUU. It's a square again because the genetic code is specific. We'll learn some more features of it, but this is protein synthesis, transcription, and translation. So let me just take a moment of time and let's kind of just expand our thinking a little bit, please. So the English language is made of 26 letters, and we use those 26 letters of the English language to form all the words that are used in that language. Well, the genetic language is only 4 letters long. The alphabet of the genetic code, when we're looking at our nucleic acid, is only 4 letters long. I think that is just so incredibly remarkable that all of life uses that same alphabet, but is so different. If you think about the same 4 letters being used over and over and over again, but we're able to produce the different amino acids that arrange in ways to make every single protein present on Earth, whether it be a spider web, whether it be DNA polymerase, whether it be a protein that our immune system produces. So that genetic language is incredibly powerful in that it only uses 4 letters, but is capable of rearranging in a way to make all of everything we see in life, all of those proteins, all of those phenotypes. Even viruses use the same genetic code. Again, they're not living organisms because they cannot replicate on their own. They don't have all the enzymes for this. They use ours. That's how they can be so small. They don't carry all their all the DNA for all those genes because they use ours. They're actually really smart from their perspective. So this is a newer version of the genetic code shown as a wheel. I'm going to show you the version that I learned with, which is shown as more as a table. But what we're looking at here are the codons and how the codons are related to the amino acids. So we've got our genetic language. I'll go through what the different layers mean in just a moment. But each codon has 3 RNA nucleotides in it. So we'll just start over here. So we've got U in position 1, and then we've got the option of the others in position 2, and then we've got the option of the others in position 3. Now, there are 20 amino acids, and that's true for all organisms. But we've also got some other codons and other material here that we'll learn about as we talk about protein synthesis. That's what these green and red pieces are. So we're going to have to learn about them. They are the start codon down here, methionine, or the stop codons. So these are signals where the enzymes know what to do. This is the table version of the genetic code, and it tells us the same thing. So we have position 1 of the codon. So that's the first piece of RNA. And then we have the second position, and then we have the third position to create that 3-member codon. So, and then we've got our start and we have 3 stop codons. So the way that we would read this is a sequence of let's just go with what we saw, U. So position 1 is a U, position 2 is a U, and then we go into here for position 3. It makes the amino acid phenylalanine, but it can be made another way. UUC also produces phenylalanine. So we do have some redundancy in the genetic code too. Every single time we get UUU, it will make phenylalanine, but we have more than one way to make phenylalanine. Leucine, we have 1, 2, 3, 4, 5, 6 ways to make leucine. So the genetic code is very specific in that every time we see the same codon, it is associated with bringing in the same amino acid into our protein chain that's being made. But there's redundancy. We have different ways of making some of these different amino acids. And some of them have lots of ways, some of them just have 2 ways. So we've got position 1, position 2, position 3 of the codon. This third position is actually pretty interesting. It's called the wobble, like wobbles over, the wobble position. because in many cases, it can be anything and it doesn't change the amino acid. So let's look in this column over here where arginine is. Position 1 is C, so we're in this row. And then we've got position 2 is G, so we're in this column. But to make arginine, we can have any amino, any of the codon, anything there. It doesn't matter what's there. We still get arginine. We could have a U, we could have a C, we could have an A or a G for the RNA nucleotides, but it still makes the same thing. So if we're talking about a mutation, something that changes our sequence permanently, if we're talking about a mutation, the best place to have it is actually in this third position, because chances are likely that it may not change the outcome. It would still have the same structure and function. We'll visit that again. Okay, so transcription is making this message RNA. There's 3 basic steps to it. The first is initiation. And that is when now we're looking at a different enzyme. It's not DNA polymerase anymore. This is RNA polymerase. It is an enzyme making a polymer of RNA because we're making RNA. We're not making DNA. We talked about DNA replication a moment ago. Now we're in protein synthesis. And yes, it comes from the same DNA strand as the template, but it's a different process that's necessary so that a cell can replicate. So RNA polymerase comes in and it binds to a certain region in the DNA called the promoter. So that's what the promoter does, is it's the site where RNA polymerase binds. And then RNA polymerase will move down the DNA and until it hits what's called a terminator, and then the process stops. RNA polymerase falls off. So if we're zooming in and kind of looking inside, we've got our pink message being made. That's the RNA. And then we've got our DNA going through. So it begins at the promoter. So we've moved a little bit. The RNA polymerase binds at the promoter, and then it moves down the template stranded DNA, making the messenger RNA along the way. So initiation is just the attachment of RNA polymerase to the promoter, which is a piece of DNA. A primer is a piece of RNA used in DNA replication. Promoter is a piece of DNA here used in RNA replication. I know it's a lot of terms. Next comes the elongation step. And that just means that we're adding in 1 amino acid at a time as each codon moves through. We're making each codon. I said that wrong. I got a step ahead of myself. We are making each codon as the DNA moves through. So here's our RNA polymerase. It began by binding to the promoter, but now we've moved down through the process. And you can see that we're having the growth of our RNA molecule, which is shown in pink here. So it's adding the RNA nucleotides in 1 at a time, following the base pairing rules off of the template DNA strand. So T to A, C to G. T, of course, being in the DNA. Our next step, the final step of transcription, is termination. And now what happens is the RNA polymerase moves all the way down till where it hits the terminator DNA. This is a sequence of DNA where the RNA polymerase falls off and then everything is done. We've got our completed strand of RNA. This is transcription. It consists of initiation, elongation, and termination. Both the promoter and the terminator are sequences in the DNA strand, in the DNA template, that allow the RNA polymerase to know like where to work between. It fills in between. Okay, let me play this short video for you.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that's transcribed into an RNA molecule is called a transcription unit. an enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Transcription. It occurs in the nucleus. Now, in eukaryotic DNA, actually in eukaryotic messenger RNA, there's some other steps that have to happen, and this is really so that the right region is made into protein, and so that our messenger RNA molecule knows or has the ability to move out of the nucleus. Now, not all of our DNA is associated with a gene or making of a protein. So the part of the DNA that makes a protein, that's a locus. We call that the gene. So exons are the part where a gene is. And the way I think about that is exons are expressed, genes are expressed. So the exons are the regions where the genes are, but in between them are other regions called introns. There is not a protein made from the intron. They're still important. We don't quite understand why they're so important, but they are important, or else they wouldn't be there. We've had billions of years of evolution. They would have gone away if that were the case. So the RNA transcript is made start to finish, but parts of it do not associate with making of the protein, specifically those introns. And then we have the cap and the tail added. These are the regions in yellow. So this is all happening in the nucleus. The cap and the tail, though, they help the sequence move out of the nucleus so that it can move to the cytoplasm and find a ribosome, or move to the rough endoplasmic reticulum. So that's what the cap and tail do. They have slightly different functions, but collectively that's what they do. So our coding sequence is put together after splicing has occurred, where the introns are spliced out, and then the exons, we have 3 in this example, they are all joined together. And this is actually one of the ways that we can make more than one protein from the same DNA sequence. It can switch out like where it's cutting, like what exons are put together is a way to get a different protein out of the same sequence. That's a way for us to save space and get more function. So exons are expressed as genes. Okay, now this thing looks a little crazy right now. What this is it's a molecule called tRNA, transfer RNA. We haven't even talked about what this molecule does yet, but it is part of protein synthesis. It is needed for translation, the second part of protein synthesis. So it's a piece of RNA, and they are shaped like a hairpin or a cross. What they carry with them, they carry the actual amino acid that is made into the protein chain. So that's where it comes from. So the transfer RNA molecules come in and they bring in the amino acids. That's when we were looking at making the protein sequence from the message, that this is where it comes from. I mean, we can't just make it up. It has to come from somewhere. Now, so the transfer RNA molecule has the amino acid called the attachment site, but it also has what's called the anticodon. So the codon is messenger RNA. The anticodon is opposite in base pair sequence to the message, to the codon. So here's a simplified form of it. All of these represent the same thing, just in different structures. We're looking at the hydrogen bonds and different structures that form it together. But a very simplified form that ties to what it functionally does is it has two ends. We've got the amino acid attachment site and then the anticodon. So DNA is DNA, messenger RNA is the codon, transfer RNA carries the anticodon. Try to write it out. I encourage you to do that so that you understand the relationship between everything. Okay, so now let's look at how the tRNA works. Now we're in the ribosome, which is the location of protein synthesis. It doesn't matter if we're talking about a free ribosome or one that is attached to the endoplasmic reticulum or even to the nuclear membrane. Every time we talk about ribosomes, their job is to make protein. That's all they do. They're actually made of two subunits, the small and the large subunit. And these are sequences that are actually highly conserved in evolution. So all eukaryotes share the same small and large ribosomal subunits. Prokaryotes have their own, and eukaryotes and prokaryotes, they do not have the same ribosomes. So the ribosome is actually part of the mystery, part of the molecular biology mystery that allowed scientists, allowed evolutionary biologists to really understand the relationship between organisms in the three domains of life. So we've got our large subunit and our small. The message goes right through it. And then there's actually 3 binding sites. Your book keeps it simplified, but there are 3 binding sites in the ribosome. I'm not going to ask you what the A site and the P site do. I'm more interested that you understand and can apply the process of translation. So what happens is these are places where these transfer RNA molecules come, and they're there. Transfer RNA molecules are around in the cytoplasm, in our cells. They can be used over and over again as long as they aren't damaged. So they come in and bring in the growing amino acid. So notice that in the transfer RNA molecule that is labeled with the letters on it, we have 1, 2, 3, 4 amino acids in our chain. But in the A site is the next amino acid to be added to the polypeptide, the P site. That's how I think of it. So the A site adds to the growing polypeptide site, just to give you some relationships between the letters there. And so now in the next step, this square is going to be added over here, and our amino acid growing polypeptide is going to be 5 amino acids long. So as the message moves through one codon at a time into the right positions, the matched tRNA molecules with their anticodon bind to the codon using complementary base pairs. A to U, C to G. That was a big mistake I just made. There's no such thing as U in RNA. And then here's a real picture of it that's been coded. These are simplistic cartoons to show it to you, but this is an actual electron micrograph of the ribosome that has been stained to show the relevant parts that you can see. So ribosomes are made from their own very special pieces of DNA called ribosomal DNA. Ribosomal DNA makes ribosomal RNA, which then is used to make these ribosomes. Again, ribosomes are very specific. They are one of those defining features for organisms that are assigned into one of the 3 domains of life. And it was through molecular biology that we really were able to work out some of those relationships, much more so than they had been understood previously. So, and now let's go through the steps of translation. Fortunately, they're the same 3 steps that we learned about for the first step, for transcription. So it begins with initiation. Here, it is the initiating tRNA molecule that binds to what is called the start codon. That was the green codon in the table we were looking at for all of the amino acids for the genetic code, there's only one start codon, AUG, that's it. And it always associates with methionine, MET, always. So proteins are kicked off with methionine. That is the amino acid associated with the initiator tRNA for everyone. Methionine can be in the middle of the growing polypeptide chain too. It's not just position 1. There's this one's unique because it's carried by the initiator tRNA. But notice the codon is AUG. So A goes to U, U goes to A, G goes to C. This is the anti-codon. It's actually got a relationship to the DNA because the DNA is used to make the codon. I'm going to say this, but I want you to draw it out on paper. The DNA and the anti-codon should have the exact same sequence with one exception. T's are in DNA, U's are in tRNA. So, and that's all because of those complementary base pairs. So we've got now, we've got the large subunit comes in and assembles and everything's ready to go. So this is all part of initiation, is the ribosome assembling, getting the initiator tRNA in site to match to the codon. And then of course we need our enzymes to come in and do the next part. Okay, so now we're in elongation, initiation, elongation. It begins as an amino acid moves over. So it's translocating over every time, moving over one step every time every time an amino acid is added on to this growing chain. And it's all by following the codon in the messenger RNA matching with the anti-codon sequence in the transfer RNA that brings the amino acids in so that they may be added to create the polypeptide. So now we've got our next one coming in. So we've got a circle at the end now, and we're adding in the square. So this is creating what we call commonly a peptide bond, which is the bond holding together the polypeptide. We talked about this when we learned about our macromolecules. And now we certainly have that square added on. And here comes translocation. Now we're ready to move everything down by one side at a time because this transfer RNA molecule is done. It already brought in the amino acid. Now it can go back and get another one and be set up and used again, but it's done for us. So everything just moves over one spot. And the process goes on until one of the stop codons is hit. So taking a look at our genetic code again, we've got 3 stop sequences, UAA, UAG, and UGA. And then we have 1 start. So these are codons I do expect you to know. I'm not going to ask you to tell me, identify other amino acids by sequence, by codon sequence, but I do expect you to know the start sequence and the, or start codon and the 3 stop codons. All right, let's go ahead and play this video for you.
The 4 nucleotides of messenger RNA are arranged in 64 3-letter words called codons. The genetic code is a language consisting of these 64 codons. 61 of the 64 codons code for a specific amino acid, while the remaining 3 instruct the ribosome to stop the translation process. Let's see how this table is used to determine the amino acid of a given codon, CAG. First, on the left side of the table, find the first letter of the codon, C. Then on the top of the table, find the second letter of the codon, A. Finally, locate the codon that has a G as its last letter, and you will see that the amino acid coded for by CAG is glutamine. Can you determine which amino acid is coded for by UCA?
All right, So just to put together the two processes of transcription and translation, which both are part of protein synthesis, transcription happens in the nucleus because we need the DNA. The DNA strand is used to make a messenger RNA molecule using RNA polymerase off of the DNA template strand. After the message is made, it moves out through a nuclear pore where it can assemble with a ribosome so that it can be made into protein. So now we have to have our transfer RNA molecule that has the amino acid and the anticodon. It begins with the attachment site of everything coming together. So that's off of the start codon. So our initiator transfer RNA with methionine is coming in to bind to that start codon. And then everything moves over one at a time through the step of elongation. And a peptide bond will form in between each of these polypeptides, creating the primary structure of the protein. It still has to fold, as we learned about in our molecule section. This continues until one of the stop codons is brought in. There is no transfer RNA molecule that matches there's no protein molecule, no transfer RNA molecule to bring a protein in for the stop codons. So that signals the termination step, which is the final step of producing the primary sequence of the protein here. Okay, so let's take a minute and look at some examples of disease and how they tie to either our DNA or our RNA or proteins, how they tie to our molecular biology. So this is just a very small piece of the sequence for the hemoglobin protein. So we're looking at the DNA, and this has the sequence CTT. It is associated with the codon. C goes to G, T goes to A, T goes to A. So the anticodon is CAA. It encodes for glutamine. So that's the normal hemoglobin. Just one base pair mutation in the DNA produces the variation known as sickle cell hemoglobin, which is associated with sickle cell disease. It's just one substitution. Instead of it being a T in the DNA, it's now an A, but that creates a U in position 2 of the codon, and it's no longer glutamine. Now it encodes for valine, and this is quite a different amino acid. So the shape of the molecule of hemoglobin changes extensively because these two amino acids have different chemical properties. So that affects its shape, which affects the function. So we do have different types of mutations, and not all of them are as risky as others, depending upon where in the codon or where in the DNA molecule associated with the codon that mutation occurs. So if we're talking about a mutation in the DNA, that has that's permanent. New protein can be made. So maybe we can make a bad protein, but what I'm trying to get at is there's a difference between a mutation in the DNA versus in the RNA. If it's in the RNA and it's not associated with a permanent change in the DNA, it's just a matter of making another protein. That's why mutations, we think of them in the DNA. That's what the definition of a proper mutation is. It's a sequence change in the DNA. Okay, so let's look at some of the examples of where and different types of mutations and how they affect. So we're still looking at, we've got our normal gene. So our DNA template is in blue, and then we've got our complementary message in pink. So AUG is methionine, AAG is lysine, UUU is phenylalanine, and then GGC is glycine. But now what happens if we have a nucleotide substitution in the place of where this G should be? We have an A. Our codon's different now. It's not GGC, it's AGC. So that is going to result in what's called a nucleotide substitution because it changes the amino acid to a completely different amino acid. Let's look at the next row down. Now we're looking at a nucleotide deletion. So now instead of it mutating and substituting, it's actually been removed. Just one nucleotide has been removed. So now we've changed the amino acid associated with the first codon where the U was removed from, but everything else after it is also changed because it's all just moved down by one. So the whole rest of the protein is going to be wrong. It's going to be out of reading frame because none of the codons are going to be the same anymore. The same happens with an insertion. So it was UUUG. Now we've got UUGUG. Now we've added in a nucleotide. It's led to a frame shift mutation as well. Everything else after it is going to be impacted because the whole sequence moves down by one position. So nucleotide deletions and insertions that are in a unit of one nucleotide or two are going to produce frame shifts because everything downstream moves. But what if it were three nucleotides of RNA that were added or deleted? That might not be as bad. If we're adding in a whole extra amino acid or taking out a whole amino acid, the potential for the rest of the protein to still be correct is good. So insertions and deletions are more impactful if it only involves one nucleotide or two. If it comes in the unit of a codon, well, maybe it will be a problem, maybe it won't. It depends if that one codon, that one amino acid, changes the shape of the protein. But certainly moving everything down frame is going to impact everything. So there definitely is some variation in the type of changes, the type of mutations and their consequences. Of course, all of this is looking at a mutation in the message. So that would just impact that protein, providing there's not something centrally wrong with a ribosome or some type of enzyme that's checking everything. So let's apply this again to hemoglobin. That's what we've been looking at. So here's our normal hemoglobin. So we're looking at the primary structure, and now we've jumped to we're looking at the different layers of folding. So we're going from primary to our secondary and tertiary structure. So that would be the alpha helices and beta pleated sheets making the secondary structure, which then folds down to give us our globular structure in the tertiary structure. And then quaternary is assembling all the chains together. And there are two of these beta chains in hemoglobin, two alpha chains. So we get the right shape hemoglobin here in isotonic media, meaning it's got the same salt concentration as our body. So this is our normal hemoglobin, but look what happens to the shape with just this mutation in one amino acid. There's a drastic shape change, and it's all because of hemoglobin. Now with valine in position 6 instead of the wild type amino acid, we get a very differently shaped beta subunit. And that changes the entire shape of the red blood cell, since hemoglobin is the main protein that's inside a mature red blood cell. I mean, in fact, mature red blood cells don't even have a nucleus anymore. They don't make protein because it's already made for them. These cells live for a very long time. Their whole job is just to carry oxygen, and that binds to hemoglobin. So this is the mutation that leads to sickle cell anemia. All right, that wraps us up. Here's your study slides. I'm going to emphasize this is really important for you to understand and be able to work with. if I give you a sequence and ask you if it's DNA or RNA, how will you tell? Also apply this back to some of the concepts we've been learning about all semester, the monomer versus the polymer. And then we've got the concepts of what we've learned in replication today, which involves DNA replication as well as protein synthesis. So this is showing us the flow of information, the relationship between structure and function, as well as replication. We're seeing quite a few of the core themes of biology just represented in this material. We've got our view of the ribosome and tying it to its structure and function. And then here is the flow diagram map to put together. All right, that completes us on, what is this? Goodness, module 14 of the semester. I hope you're well, that you're safe and happy. Take care, everybody. Reach out by email anytime you need me. Bye-bye.